Welcome to our webinar about the effect of different SARS-CoV-2 variants on COVID-19 assays and vaccines, organized by Antibodies-online in cooperation with Nanotemper technologies. My name is Stefan Pellenz, Product Manager at Antibodies-online, and I will be presenting today together with Andreas Lange, the Senior R&D Scientist at Nanotemper. This installment is an update of a webinar presented 3 months ago at the end of June. For those listeners that have already attended the previous presentation, I would like to note that the provided background information has remained largely the same, with the addition of new findings of the last months. Beyond the content of the previous presentation, we will be showing additional data that we have produced over the course of the summer. The webinar will be recorded and we will make it available on our website within the next week. Any personal information about participants will be removed, so don't hesitate to use the chat window to ask any questions that will be answered in the Q&A session subsequently to the presentation. Let me give you a short overview of today's presentation. After a few introductory words about today's organizers and speakers, I'm going to talk a little bit about SARS-CoV-2 proteins and in particular the S protein and its significance in context with existing vaccines. Subsequently, we will take a closer look at the S protein structure and the effects of some of the mutations in virus variants of concern, and I will provide some background on some of one exemplary anti-S protein antibody. Andreas will then present detailed affinity data that we have produced for this antibody in context with the S proteins from different SARS-CoV-2 variants, as well as for some other antibodies. After putting this data in perspective to some real-world data, we conclude the presentation with some summarizing remarks and the aforementioned Q&A session. Organizers of today's webinar are Antibodies-online and Nanotemper's technologies. Antibodies-online was founded in 2006 as a spin-off of Aachen Technical University. We are now located in Aachen, Germany, and in Philadelphia, Pennsylvania, in the US. Our mission is to make the search and purchase of antibodies, proteins, kits, and other proteomics products easy and straightforward. We count more than 6,000 institutions in 65 countries as our customers, some of which are indicated here. In April 2020, within 1 month after COVID-19 had been declared a global pandemic, Antibodies-online offered 600 SARS-CoV-2 related antibodies, proteins, and kits. By May 2020, many of these had been delivered to customers in over 25 countries. And by now, in September 2021, we have delivered an ever-increasing number of products to more than 500 labs. This confirms Antibodies-online as a trusted source for COVID-19 research. Beside great products, we strive to provide right content relating to the research fields these products are being used in. We have, for example, released the world's first product-centric SARS-CoV-2 mutation tracker to help our customers to keep pace with the highly dynamic COVID-19 research. We have also compiled a SARS-CoV-2 handbook providing an overview of the SARS-CoV-2 virus, COVID-19 biology, and related products. The mutations tracker and the handbook are both freely accessible on Antibodies-online. Validation data plays a key role at Antibodies-online, and so we pick and recommend the products that come with the best data. We do also generate additional data through our independent validation program. The data we will present today fits into our data mantra as we go beyond what you usually get to see. Nanotemper Technologies was founded in 2008 as a spin-off of Ludwig Maximilian University in Munich. The company is based in Munich, Germany, and has by now 8 additional offices in Europe, the US, China, India, and Japan, with a total of approximately 160 employees. The product portfolio comprises 4 different product lines for the characterization of proteins. The monolith and the Anthos platforms, which use the effects of microscale thermophoresis and temperature-related intensity change to measure molecular interactions, and the Prometheus and Tycho platforms, which apply nanodifferential scanning fluorometry to measure protein stability. All in all, more than 1,000 Nanotemper instruments are used around the world and were cited in more than 5,000 publications. Our speakers today are Dr. Andreas Lange and me, Stefan Pellenz. Andreas holds a doctorate in biophysics from the Technical University of Munich and has extensive experience in protein interactions. As application scientist, Andreas has advised Nanotemper Technologies distributors in Japan, India, and South America on the use of the different Nanotemper instrument platforms. He is now senior R&D scientist at NanoTemper Technologies. I have obtained a Ph.D. in genetics from Université Paris 7, followed by a postdoc at the University of Washington in Seattle, Washington. Both during my Ph.D. and postdoc, I have worked with engineered DNA endonucleases, and I have therefore a long-standing interest in protein structure and how it influences protein function. I'm now working as a product manager at antibodies-online, and I'm responsible for our independent validation program, which brings us to today's topic. SARS-CoV-2, the causative agent of COVID-19, has a positive sense single-stranded RNA genome with a size of almost 30 kb. The genome includes a total of 26 proteins. 16 of these proteins are so-called non-structural proteins. That means they do not contribute to the structure of the assembled virion. Their functions are primarily downregulation of host protein production, virus replication, and the production of new virions once SARS-CoV-2 enters a host cell. Among these are the host shutoff factor, NSP1, the two virus proteases, PL-Pro and the main protease C3L-Pro, and the RNA-dependent polymerase and the exonuclease for proofreading during genome replication, the main reason for the comparatively low mutation frequency of SARS-CoV-2. In addition, the SARS-CoV-2 genome contains sequences for 9 accessory factors whose function in virus replication, modulation of the host's immune response, and packaging remains largely unclear. Lastly, the genome encodes for structural proteins, the spike, envelope, membrane, and nucleocapsid protein that makes up the that make together up the assembled virion. For this presentation, I will mainly talk about the S protein shown here in orange. So why the focus on the S protein? So here, what you can see here is a list of several SARS-CoV-2 vaccines that have been approved until now for use in humans. They rely on different technologies. The BioNTech, Pfizer, and Moderna vaccines built on RNA encoding a viral protein packaged in lipid nanoparticles for delivery. Other vaccines, like the also well-known AstraZeneca, the Sputnik V vaccine, and Johnson & Johnson's Janssen, and the CanSino Biologics vaccine, rely on delivery of viral S protein DNA and subsequent production of the antigen in infected cells. While the delivery of the genetic information for the antigen varies between these vaccines, they all rely solely on the S protein to mount an immune response against SARS-CoV-2. The Novavax and SARS-CoV-2 vaccine are more recent additions, which are based on virus protein antigen and this is actually a first for any vaccine, circular DNA encoding the antigen respectively. In spite of the different platforms, the S protein is also the pertinent antigen for both of these vaccines. The two vaccines shown here on the bottom of this table, the Sinovac Biotech vaccine and the Indian Covaxin, use inactivated SARS-CoV-2 virions. So therefore, they display also the other structural proteins as opposed to only the S proteins. However, the S proteins appears to be the most immunogenic of the SARS-CoV-2 structural proteins, and one can assume that even in these whole virion vaccines, the S protein plays a crucial role to elicit an immune response. Besides, it has to be noted that the S protein is also the host's first contact point with the SARS-CoV-2 virus. So in short, the S protein is very important for all COVID-19 vaccines, independent of the selected platform. It is also one of the most relevant targets for antibody-based SARS-CoV-2 tests. In order to better understand the body's interaction with the SARS-CoV-2 virus, it is helpful to have a closer look at the S protein structure. The S protein protrudes from the spherical virion, shown here on the left, and gives it its characteristic appearance resembling a crown that has earned the group of coronaviruses its name. Zooming in on the S protein, here on the right, shows that it's actually a homotrimeric complex consisting of 3 monomers, shown here in yellow, green, and yellow, gray, and green. The lower S2 section of the complex contains the trimerization interface and the membrane proximal stalk that is important for membrane fusion and virus entry. The upper S1 section contains an N-terminal domain, or NTD, and the receptor binding domain, or RBD. The three RBDs have to transition from closed conformation here in the middle to an open position, shown for one of the RBDs here on the right, the yellow one, for the virus to contact the ACE2 receptor and the host cell surface, on the host cell surface. While the antibody has been shown to contain many immunodominant regions eliciting a T cell response, the RBD is thought to be the primary target for antibodies. Therefore, mutations within the RBD, shown here in orange, are highly relevant for the effectiveness of antibodies, either in the host's immune response or immunoassays. Having a closer look at S proteins present in SARS-CoV-2 variants of concern and variants of interest revealed several amino acid changes that can potentially affect binding of antibodies, recognizing the canonical RBD sequence. Especially the mutation E484K stands out because which is here, because an acidic, negatively charged glutamate has been changed to a basic, positively charged lysine. Biochemically, this is a significant change, and this is consistent with the observation that variants containing this mutation, like, for example, B1.351 beta and the P1 delta lineage, reduce the effectiveness of treatment with serum from convalescent patients, not parents. It is also to be expected that some of these mutations might negatively influence the function of some existing antibodies used for SARS-CoV-2 lateral flow assays, Elisas, and other immunoassays. In order to verify the usability of any antibodies targeting the S protein, one has therefore to test if they retain the ability to bind the intended target in novel virus lineages. So shown here from the left to right are structures of S proteins from SARS-CoV-2 variants of concern B.1.17 alpha, B1.351 beta, P1 gamma, and P1.612 delta. For clarity's sake, the amino acids that differ from the S protein of the canonical Wuhan wild-type strain are indicated here only for the gray monomer. Many of the SARS-CoV-2 variants of concern mutations appearing in the daily press are located within the S protein's RBD, so, which is shown here. These are, for example, E484K in the beta and gamma variants, or L542R and T478K in the delta variant, which is presently the most concerning virus variant. This is consistent with the observation that S proteins from variants that are more readily spread have an increased propensity for the RBD upstate and show increased ACE2 receptor binding. Additionally, mutations like K417N, E484K, and N501Y have also destabilizing effects that can negatively impact binding of neutralizing antibodies. Production of neutralizing antibodies is one aspect of immunity upon vaccination or infection with SARS-CoV-2. Simply put, neutralizing antibodies get in the way of the S protein's interaction with the ACE2 receptor on the surface of the host cell. The main target for neutralizing antibodies is, as I had mentioned before, the S protein's RBD, shown here separately on the right. Depending on the location of their targeted epitopes, neutralizing antibodies are classified in classes 1, 2, and 3. An antibody attached to these areas of the RBD interferes with binding of ACE2 to the ACE2 host cell receptor. Of particular interest are the class 2 antibodies, indicated here in yellow, because these antibodies effectively block the receptor binding motif, or RBM. This motif consists of amino acids that form direct contacts with residues on ACE2. And here one finds, for example, the residue 484, and it has been shown that E484K mutation can effectively abolish binding of class 2 neutralizing antibodies, thus negating their neutralizing effects. In contrast to the class 1, 2, and 3 antibodies, there's a fourth class of anti-S protein RBD antibodies, indicated here in red in the lower left corner. These class 4 neutralizing antibodies are characterized by the fact that they bind in none of the 3 sites of the other 3 classes. They do also not contact any residues that bind directly to ACE2. One of these antibodies is the antibody clone CR3022. CR3022 was originally isolated from the serum of a convalescent SARS-CoV patient about 15 years ago. The antibody on its own shows only weak neutralizing effects on SARS-CoV-2 since it binds to a conserved epitope that does not interfere with ACE2, as I had just mentioned. It does, however, synergize well with other neutralizing antibodies. That means the combined effect of the two neutralizing antibodies is larger than would be expected. Besides the function as a neutralizing antibody, CR3022 is frequently used as reference in immunoassays targeting SARS-CoV-2 S proteins. Shown here on the right is a schematic representation of the antibodies epitopes in yellow within the S proteins RBD, shown here in orange. Mutations are indicated in the rows above for each of the variants of concern in gray, or variants of interest up here in green. Based on the RBD's primary sequence, the epitopes of antibody CR3022 do not include any of the amino acids mutated in any of these variants. Unless the quaternary structure of the respective variant's S protein trimer has an effect on binding of CR3022, we hypothesize that the antibody should still effectively bind all of the S proteins. In order to test this hypothesis, we sourced high-quality, full-length trimeric CoV-2 S proteins, including the membrane proximal stalk from our protein expression lab here in Germany. The S proteins tested were those of the canonical Wuhan wild-type strain, the S proteins from variants B.1.117 alpha, B.1.351 beta, P1 gamma, B.1.617.2 delta, and then the variants of interest B.1.429 epsilon, B.1.525 eta, and B.1.617.1 kappa. The different S protein variants used, plus this year's 3022 antibody itself, are all available through our catalog under the product numbers indicated here on the right. For the measurements, we teamed up with Nanotemper technologies, and Andreas will show you on the next slides whether our hypothesis regarding binding of different S proteins variants by antibody CR3022 held true or not.
Yeah, thank you, Stefan. So our goal at Nanotemper was to really measure the binding affinity between this CR3022 antibody and these different S protein variants that you can see on the right. And to do this, we used our monolith benchtop instrument, which is displayed here in this small picture, which uses microscale thermophoresis or MST technology to measure dissociation constants between two molecules. So we want to measure the KD between two molecules with a has to be fluorescent or fluorescently labeled. And in our situation here, it made a lot of sense to label the antibody and then use all these S proteins as unlabeled ligands. Our second generation NHS labeling kits, which labels lysine residues on the antibody. Next step then is to prepare a dilution series of the unlabeled ligand. Here we went for a concentration range between 7.6 picomolar and 250 nanomolar, so that we were also able to see if there's binding maybe only in the higher nanomolar range. This dilution series then gets mixed with the fluorescently labeled antibody, and we had here a concentration final of 2.5 nanomolar of the antibody. This was possible because we used our instrument with picosensitivity, and this then allows you to also measure KDs that are even in the picomolar range, so really strong binding interactions. On the next slide, you can already see the results of these measurements. These are so-called dose response curves. We have here on the left the F-norm signal, that's the primary signal from MST. And at the bottom, we have the concentration of the S protein, starting at picomolar concentrations here and going up to high nanomolar concentrations.
But since we were mainly interested in relative differences, we wanted to see which of the S protein variants is bound strongest by the antibody. It's best to look at this data in the normalized view, and this is called the fraction bound plot.
So these curves now, they are sigmoidal, they start at 0, there's no binding, and they end here at 1, which means 100% binding. And basically, the more these curves are shifted to the left, the stronger the binding is, the more they are on the right, the weaker the binding is. From this, we can already see that the curves look different. So it's not like the antibody binds all the variants equally, but there are some differences. For example, the canonical version, that's the red curve here, that was the first one we measured, that showed the strongest interaction. We got here a KD value of around about 1 nanomolar. That's a really good KD for an antibody. A lot of commercial antibodies are, for example, in the micromolar range, so a factor of 1,000 weaker. If you have a really, really strong antibody, it can be maybe a couple of hundred picomolar, maybe a couple of tens picomolar even. The thing we saw was that the B.1.1.7 variant behaved very similarly. So the KD here is not much changed. And this was a reoccurring picture that I got when I did more antibody measurements. I always found that the B.1.1.7 behaved very similar to the canonical S protein. The surprise was then B.1.351, which showed a binding that was a factor 10 weaker. So here, even though, as we just heard, the epitopes of this antibody are not affected by the mutations, still we see here in effect, antibody can only bind a little bit weaker to the S protein. Then some other variants again showed a bit stronger binding, but I think the most interesting one here is the blue curve, that is variant, so B.1.617.2, which showed a KD of only 50 nanomolar. So we have a factor of 50 weaker binding than to the. I think this is really, really interesting to see that the Delta variant apparently here makes the binding much weaker. Nevertheless, the KDs all are still nanomolar, so it's still very good, which means that this antibody can, for example, be used in Elisas or in lateral flow assays or in neutralization assays without any problems. So it will still bind to all the variants. In comparison here, we have also the KD between ACE2 and the S protein, and that was found to be around 15 nanomolar. So you can see we are in the same range. Some antibodies can bind even stronger, but in some cases, the binding to the antibody can become weaker. And then, of course, the ACE2 will be the preferred target for the S protein. Next, we looked at two more monoclonal antibodies. One was called SP422. And that one was specifically raised against the S1 subunit of SARS-CoV-2. So here we expected to see strong binding to at least to the canonical S protein to which it was raised. And that was confirmed. So we got here a KD that was even picomolar. Also for B.1.1.7, we found very strong binding. For 3 more variants we tested, we found slightly weaker binding. Still a low nanomolar KD, so still very good, but in comparison, it's maybe a factor of 5 weaker. So there is an effect also for this antibody. The B.1.617.2 then again bound quite strongly. So I would say here comparable to the canonical and the B.1.1.7. So here there's no influence of the mutation. Second antibody then was a big surprise. There was an antibody called SP185. Here again, a very strong binding to the canonical S protein and the B.1.1.7, about half nanomolar KD. We did not see binding anymore to the other 3 variants. So you can see that the curves, they only increase very late. You need high nanomolar concentrations to get something to interact. So we can estimate the KD to be maybe in the really high nanomolar, maybe low micromolar range, but there's no binding in the range here between 1 and 10. Again, bound a bit stronger. Again, a factor 5 worse than to the canonical S protein. You might be wondering how MST is different to other methods that are used to study antibody-antigen binding. And 2 very popular ones are SPR and ELISAs. So in this short method comparison, I would like to tell you a bit what are the similarities and the differences. First, a similarity, that's the approach. So in all 3 methods, we have a titration approach. This means that we have one molecule that is tested at increasing concentrations, and then we try to see what concentrations do we need to get an interaction. That's the same for all these three methods. What is different is the readout. MST and SPR are quantitative measurements, which means that we really get here a KD value as a result. The KD is a hard number that lets you compare different ligands, different targets. So if you do two measurements, you get two KD values, and then you can say this one binds stronger than that one. With ELISA, it's not that quantitative, it's rather semi-quantitative. Here, an enzymatic reaction is followed, so you get kind of a qualitative answer if you have an interaction or not. It's not that easy to compare two different ligands, for example. Regarding immobilization, MST is an immobilization-free method. It's in solution and fluorescence-based. In SPR and ELISA, there is a surface, so you have to immobilize a molecule to the surface. In SPR, this is done usually site-specifically. There are different chemistries available, different chips. SPR chips are used. In ELISA, it's a non-specific adsorption to plastic. And this can be a drawback of the method because not all proteins tolerate that they are non-specifically adsorbed. So it can happen that you have denaturation of the protein when it's sticking to the plastic. Regarding fluidics, in MST, there's no fluidic system needed because we measure already in solution. In SPR and ELISA, you need to have a way to exchange the liquid on top of the surface with the binding partner. In SPR, this is done via a fluidic system, a flow cell. In ELISA, you do it with multiple washing cycles. This can be another drawback because if you imagine you have a weak binder and you have to do washing to exchange the solution on top of the immobilized binding partner, then weak binders can dissociate already while you're washing, and this means you can miss them. So you will not measure an affinity that is maybe high micromolar. In MST and SPR, we are always on equilibrium, so we can achieve equilibrium conditions, which means we can also measure affinities that are, for example, millimolar. So you have a really wide range of addresses. Finally, regarding sample consumption, in MST, we measure in small glass capillaries, which take around 10 microliters volume. And that's really beneficial if you have, for example, a ligand where you want to have high concentrations, but you don't have a lot of sample, a lot of volume, then it's good that you only have to use 10 microliters per concentration. Volumes in SPR and ELISA are maybe slightly larger. And finally, the time to result. In MST, you have to do the labeling. That typically takes around 1 hour. But once you have your molecule labeled, you can prepare, for example, aliquots. You can store them frozen. And then whenever you want to do a new titration, you can just take a fresh sample from the freezer and run it. And it takes maybe about 10 minutes to do the measurement. In SPR, it's maybe a bit longer, the immobilization, then the titration. And ELISA probably is the longest method and the most hands-on method because you have all these multiple incubation steps, washing steps, and surface blocking steps. And finally, in this slide, I would like to show you how MST compares to ELISA. We were very lucky that antibodies-online also performed in parallel interaction measurements with ELISA. And you have here 4 graphs. Left is MST, right is ELISA. The two antibodies that I showed you on the slide before. And yeah, you see already the curves look quite similar. And that was really nice to see that we obtained the same results with these two methods. So we could also see in ELISA that these two variants of concern here bound weaker, but they still bound. While for the second antibody, also in ELISA, these did not bind at all. And I found this really remarkable because we had completely different assay orientations in these two methods. So it's not only two different techniques, but also how we set up the assay was different. In MST, we labeled the antibody and we titrated the S protein. And in ELISA, the S protein was immobilized and the antibody was titrated. And if you still then get the same results here, this is a really powerful sign that the data is all real and significant. And you can really draw conclusions on that. For example, here, these variants B.1.351 and P1 are bound weaker than the. And with this, I give back the word to Stefan, who will now tell you how the MST data that we got compares to real-world data.
Yeah, thanks, Andreas. As I mentioned before, our primary interest for the experiment Andreas just described was to find out whether antibodies used for immunoassays can still bind S proteins of novel SARS-CoV-2 variants. Naturally, this question is also highly relevant for antibodies produced in response to SARS-CoV-2 infection or following vaccination. A quick comparison to some real-world data shows that the effect of the S protein mutations is indeed measurable. So here in this first study that came from South Africa, the authors came to the conclusion that the variant of concern B.1.351 beta does indeed escape neutralization by class 1 and class 2 antibodies. In the second publication here from Israel, the BioNTech Pfizer mRNA vaccine was shown to efficiently neutralize the SARS-CoV-2 wild type as well as the B.1.1.7 alpha variant. In comparison, neutralization was almost reduced sevenfold with the B.1.351 beta variant, which is actually remarkably similar to the results we got with just one monoclonal antibody, as opposed to a vaccine response where you will be seeing a mix of different antibodies. The third article here from France concludes that the B.1.617.2 Delta variant can escape antibodies targeting epitopes both within RBD and outside of this domain. So here they were actually using monoclonal antibodies as well as a reference. In this last series of publications from the UK, the authors looked at the influence of different SARS-CoV-2 variants on the neutralization effect of sera from convalescent patients infected with the Victoria strain, which is very similar to the original SARS-CoV-2 wild-type strain from Wuhan. The authors also checked the effects of post-vaccination sera and sera from patients infected with different SARS-CoV-2 variants of concern. To illustrate the potential influence of different SARS-CoV-2 variants, I'm focusing here on one single aspect, which is the neutralization by sera from convalescent patients. While there seems to be no widespread escape of the B.1.1.7 alpha variant, the sera's neutralizing effect is about 13.3-fold reduced for variant B.1.351 beta and 4-fold reduced for the B.1.617 delta variant. The effect of neutralization by different antibodies varies depending on the respective epitopes, similar to what we have seen on our measurements. This is just a word too, that the test that I did here as well with monoclonal antibodies. It is still not clear what level of antibody production can be considered a good response to SARS-CoV-2 infection or to vaccination. So a decrease in neutralization does not necessarily equal a failed immune response. In addition, antibody production is not the immune system's only means to counter an infection. I mean, there is a T-cell response as well, which has been shown to be very important. However, it is notable that depending on the antibodies produced, mutations in the S protein can really have significant effects. So to summarize our findings, based on the MST measurements, the neutralizing antibody CR3022 recognizes the SARS-CoV-2 wild-type S protein, as well as S proteins from the alpha, beta, gamma, delta, epsilon, eta, and kappa variants. This means that although the binding affinity of SARS-CoV-2 antibodies to their antigens may be affected by mutations, they can remain valuable tools also for novel variants, depending on the epitopes they bind to. However, we have also shown that it's essential to verify that antibodies used in immunoassays retain their ability to detect the intended antigen in novel virus lineages. So effectively, this has to be verified for every antibody for each variant if one wants to use it in such assays. We are now working on extending these binding measurements to S proteins from other SARS-CoV-2 variants, namely the B.1.621 variant of interest. And besides, we are already testing additional monoclonal and polyclonal antibodies as well as different antibody platforms in comparison. More generally, we at Antibodies Online are constantly extending our offering of SARS-CoV-2 related antibodies, proteins, and assay kits. So stay tuned. And this concludes our webinar today. Thank you very much for your interest and your attention. In case any questions or interest in any of our products mentioned here beyond is coming up, please don't hesitate to get in touch with either of us, either by email or by phone, and we'll be happy to take your questions now.